Good afternoon, everyone. Hope you can hear and see me perfectly well. My name is Martin Westgate. I'm team leader for Science and Decision Support at the Atlas of Living Australia, and welcome to our August webinar for August 2022. Before we start, I'd like to acknowledge the traditional owners of the land that we're broadcasting from, which in my case today is the Ngunnawal people in Canberra. And I'd like to pay my respects to their elders past and present. We do, however, have speakers from many parts of the country today, and they'll perhaps introduce the countries they're speaking from in due course. So as many of you may know, it's Science Week, which is always a lovely time of year, time to celebrate the achievements of our scientists and researchers from around the country. And so normally in these sorts of webinars, we focus on the sort of discoveries that people make and the process and the work that they generate, and that's wonderful, and it's amazing to see those discoveries and communicate them to a wider audience. But I think what often gets lost in those conversations is the people that make those discoveries are real people, and also I think that it's sometimes hard to know what a scientist does day to day. You know, if you go to an office or many other jobs, people have a perception of what that's like that is lacking for scientific work, which is in many ways a bit mysterious to a lot of people, I think. And so today's, to celebrate Science Week, we're hearing today from three scientists who are going to talk about their careers to date and how they've made the careers that they now inhabit and what their choices were along that way. And I'm really quite excited to share those journeys and hear from those speakers. So in order that we'll be hearing from them today, we've got Mel McKenzie from Museums Victoria. Give us a wave, Mel. Got Sandia Balasubramaniam, who's from here at the Atlas of Living Australia, and Casey Gibson from University of New South Wales. Thank you everyone for joining us, and we're really very excited to hear your talks today, starting with Mel. Mel, you have some slides, I believe. Would you like to start to share your screen?
Sure.
Lovely, that's working perfectly. So over to you, Mel. Thank you so much.
Great, thank you so much. I'd like to acknowledge the traditional owners of the land on which I live and work, which is the Woiwurrung, Wurundjeri and Boonwurrung peoples of the Eastern Kulin Nations, and pay my respects to elders past and present. And I also would like to acknowledge that the First Nations people are the first scientists, which is pretty exciting too. You'll see from this image here that I work at Museums Victoria, and in particular at Melbourne Museum. Now, for any of you who've been to Melbourne, you may have been to the museum and you may think of it more in around its exhibitions. So if anyone's been lucky enough to go recently, you would have seen the amazing Triceratops and possibly this new exhibition Charmer as well. But we're not just exhibitions. There's actually a huge Museums Victoria Research Institute behind the scenes. Now, this is a lot of different collections and a lot of different researchers and scientists working really hard behind the scenes as well. And I am lucky enough to work in one of those collections. So my collection is the Marine Invertebrates collection, and we have a very big collection, over 1 million specimen lots, so over 5 million specimens. And basically what that is it's a life library. It's specimens in jars on shelves, and those specimens have been collected in various ways over many, many years. Some of the animals don't actually fit in jars, so you'll see that in our collections we have everything from tiny tanaids to giant squids like this one here that Lou's holding up. And our collections are used by a variety of people. So of course, people like the public via exhibitions and collections online and things like that. But we also even have artists and designers and historians and lots of different people come through to use the collection. But most of all, of course, is researchers. And our researchers use the collections in lots of different ways. So, of course, one of the main things is to describe and classify new species. And within our collections, we actually do have a lot of new species just sitting there waiting to be described that have been collected over many, many years. So if there's any budding taxonomists out there, please, please, we need more taxonomists to actually help us to describe those species, because without knowing what we have, it's very hard to see what we might be losing or what might be changing. But these collections are also a great record of biodiversity over a long period of time. So they help us to understand how habitats and ecology have changed and what the effects of something like climate change might be. They're also very important for things like marine pests. So you'll see just here we have the Asterias there, the Northern Pacific sea star. So being able to identify pests versus native animals, and of course now more and more things like biosecurity, eDNA and things like that. And of course, medicine, bioengineering, so many things. And we do use the collections to answer the really big questions from evolution to extinction. And for any of you who were watching some of the ALA seminars in the past, there's been some fantastic talks on the use of historic collections for this. So every animal is basically a data point, or it will be. So all the animals that go into our collections, we try and capture all the data that goes with them. So where they were collected, when they were collected, you know, and anything about them at the time. That goes into our database that then feeds out into other databases like the Atlas of Living Australia. So how do we use our collections? Well, we use them for, how did we collect our collections? Some of them were collected historically by purchase. We had exchange and of course donations, and more recently things like fieldwork. So day in the life of a collection manager. I have the most amazing job, I have to confess. I love it. So my career is really is multitasking. So on any given day, I can be doing anything from topping up a jar of squid to packing a worm to send to the Smithsonian, to choosing a crab to go on display, giving a talk, illustrating a sea cucumber, databasing, anything really. So there's just so many fantastic jobs as a collection manager that I get to do. Oops. And I also have a special research interest in sea cucumbers. So these little guys here, very beautiful, I think. So when I get the chance, I do try and identify different sea cucumbers too. And sometimes I do get to leave the office, which I get very excited about on fieldwork. One of the first great trips that I got to do was to go to the Falkland Islands on a Shackleton scholarship. So when I went to the Falklands, I actually went to help them with collection management of their marine invertebrate collection, but I also got a chance to study sea cucumbers there. And in my free time on the weekends as a volunteer, I got to volunteer for the Millennium Seed Bank and also for a penguin isotope study. So yes, there I am, dressed in very nicely padded astronaut clothing, trying to tackle Gen 2 penguins. And back in Australia, I've had the chance to go on many bush blitzes and bioscan surveys. So collection managers and other researchers in the department go out with different institutions to do these mini snapshot surveys of different places. Sometimes that can be for bushfire recovery or for many different reasons, but just to get an idea of what the animals are in that area. And I've also been lucky enough to go on 3 Australian and 3 Antarctic voyages. So the most recent of the Australian voyages was to the Indian Ocean territories, where I went last year. Now, every voyage that you go on, look, I have to confess, they very rarely run smoothly to plan. So you can see there that the blue line is the expected voyage and the expected sampling, and the yellow is the actual. So with COVID, with delays, with getting called back to port, with being not allowed into Western Australia, we didn't get our whole trip done, but luckily they're going to let us go out again at the end of this year. But it was still a voyage of discovery, and we did get a lot of mapping and sampling done. And that's actually really important because that feeds into management of those areas. And around Christmas and Cocos, we've just had 2 new Australian marine parks announced. So it's really important that Parks Australia knows what's there so that they know how they can manage it. And we found some great stuff under the water. We found some undersea volcanoes. The thing that looks like a dog there is actually Christmas Island. It's at the top of a seamount, which is really cool. And of course, we collected lots of samples. So we were sending these sleds and nets and water bottles, these little sort of modified research nets, all the way down to the bottom of the ocean. So sometimes that can be 6 kilometres deep to do a mini trawl to see what's down there. And when everything comes up, oh my goodness, the group of scientists, as soon as they see that the nets are on the deck, everyone is just so excited, just not sure what they're going to see. And then we bring everything back to the lab. And in this case, because we're in the tropics, we had to sort on ice. So you can see that we basically just sorting like with like, identifying what we can, photographing what we can, then pickling and databasing everything so that we can then take it back to the museums for more people to identify it further. And there were so many cool critters. I mean, I love things like the cookie cutter shark, this fang-toothed fish. I'm not going to mention some of the other ones that have rude names, and some beautiful invertebrates. And even cooler, collecting in Antarctica. So as I said, 3 different voyages. The most recent one was to the Larsen Sea ice shelf. And getting there is never actually easy. Usually you have to go through the Drake Passage to get to Antarctica if you're going from the British side. And that's always quite rough. But the first iceberg is definitely worth it. It's absolutely stunning when you see your first one. And then you reach the nice, smooth calm of the ice flows, which is just beautiful. And really gorgeous light down there too. And of course, much like with the Australian voyages, we're sending equipment down to the bottom of the ocean, but quite different from working in the tropics. Here you're working in 7 layers of clothing, and you're worried about the animals freezing to the nets compared to them rotting. And you can see down there, that's me and Helena trying to retrieve some specimens. And there we are again, just picking specimens off the net before they freeze. And there's just a few of the animals that you might see down there. I mean, a lot of them, it's the first time they've ever been seen really by humans. So it's so exciting to be at the forefront, I guess, of that kind of thing. And of course, there are some gorgeous animals above too, like this little fat little seal and minke whales and fur seals and these gorgeous Adelie penguins. So, as you can tell, I really love my job, especially the fieldwork, but I love everything about it. So a little bit about how I got here. Lots of different ways. So study, travel, and many mentors. I loved going to the beach as a kid, so I did love looking in rock pools, especially for sea stars and things like that. But it wasn't actually until I got to university that I decided to sort of go down the zoology track. So originally I was thinking, oh, vet, journalist, doctor, something like that, but I was very indecisive. So I went for a Bachelor of Arts and a Bachelor of Science. And I'm really actually happy I did that, because the art side meant that I got to do some really different things like history and English and just other things on the side while I was also studying zoology. And while I was there, I had a great tutor, an ALA tutor anyway. Ali Wallace actually helped me get through the first year of science. And she also brought Mark Norman into one of the labs one day, and he told us all about these research collections at the museum. And I got very excited and decided I wanted to volunteer the very next day. And luckily I got to volunteer with his supervisor, Lou. And what I was doing with Lou was counting suckers on these very cute little pajama squid. So I got to count lots of suckers for him, and then eventually I was in the right place at the right time when jobs came up. So I helped with relocation of the museum, and then I became an assistant collection manager, and then I actually went on a completely different track. I actually went to Japan for 7 years, where I ended up teaching English, and then I taught the teachers, and then I helped develop the curriculum and write children's books and do all that kind of thing. So when I came back to Australia, I actually spent a couple of years in publishing. But that whole time I still really wanted to get back into science. So I volunteered, and I volunteered with this mentor down here, Mark O'Loughlin, who was really into sea cucumbers at the time, and that's how I fell into that field. And then again, I was in the right spot at the right time when a collection management job came up. And ever since then, I've been supported by wonderful mentors, led by Joe Taylor. The collection management team's been great, and that just means that I've always wanted to stick with this job, because it's just always been fantastic. So there's the images. So thank you very much, and I look forward to hearing your questions a little bit later.
Lovely, thanks, Mel. That was a wonderful overview of the work you've done, which sounds extraordinarily diverse. I've got to admit, as someone who saw my first sea cucumber as an adult, and
you're missing out.
I was not as impressed with it as you might have been.
Oh, come on.
I mean,
they're beautiful on the inside.
I didn't get that far, I've got to say. But no, that was wonderful. And one thing I forgot to mention at the beginning for all those watching at home is that there is you should see in your browser a little icon that enables you to ask questions, and those will come through to the speakers. We've got time for questions and discussion at the end, so pop any questions you've got in the chat, and we will we'll endeavour to get to as many of them as we can. There's also a little poll in there so that tells us about who you are and where you're dialing in from today. So if you want to fill that out, feel free. As I said, we'll keep our questions largely to the end, Mel, so I won't query you too much further. But our next speaker is Sandhya Balasubramanian from here at the Atlas. Sandhya, would you mind sharing your slide?
I would love to.
Lovely, thank you so much. Lovely. I'll hand over to you. Thank you, Sandhya.
Thanks, Martin. Hello, everyone. I'd like to start by acknowledging the traditional owners of the lands on which I live and work, the Wurundjeri people of the Kulin Nation, and to pay my respects to elders past and present. I recognise the spiritual and cultural significance of land, water, and all that's in the environment to traditional owners, and their continuing connection to country. My story starts a very long time ago, when I moved to Melbourne to study zoology at university. I became really interested in animal behaviour, and so I applied for an honours project on the incubation behaviour of black swans. This meant that I spent about 6 months trying to sneak my hands under the bums of swans to check on their eggs while they were nesting. This was my first taste of fieldwork, and I realised it was something I really enjoyed. I also really enjoyed the process of developing a research question and working through the steps to get the answer to that question. On the basis of this, and not very much else, I applied for a PhD. My PhD was my first taste of really intensive fieldwork and research. I was looking at how differences in habitat fragmentation affected immune genes in a suite of woodland bird species. This meant that I spent the first 2 years of my PhD surveying 60-odd sites in the box ironbark forests of Victoria. By the end of this period, I had sampled over 1,000 birds from more than 20 species. For someone who wasn't familiar with country Victoria, it was a really great introduction to the landscapes and species in the region. It was during this time that I developed skills in identifying native birds, as well as a newfound love for the sound of magpies caroling at dawn. My work in this area added to our understanding of the evolution of immune genes in Australian birds, which up until that point had been largely unstudied, as well as showed how differences in habitat quantity and composition can have really stark differences on the health of woodland species that depend on complex habitat structure. After I completed my PhD, I worked as a postdoctoral researcher at Museums Victoria for about 3.5 years. I received a research fellowship that gave me free rein to explore my interest in threatened species biology. It was really great fun, and I worked on a few different projects during this time. These are some of the species that I was lucky enough to work on. On the left is the plains wanderer, the hooded plover in the middle, and the bushstone curlew on the right. These species are all threatened to varying degrees, and the threats that they face are all different. But the thing that they have in common is that all the projects aimed to improve the management strategies for the species using genomic data, which is how I got involved. For instance, the plains wanderer is one of the most threatened birds in the world. They're really small birds. They're only about 15 centimeters tall, and they weigh between 50 and 100 grams. To put that in context, males weigh about 2 and a bit Tim Tams, and females about 5 Tim Tams. In many species, males are much more brightly coloured, but this is reversed in the plains wanderer. That's the female on the left, and you'll notice she has a really lovely orange bib and the monochromatic necklace, whereas the male is on the right, and he's a lot more cryptic looking and blends into the landscape a lot more easily. These birds are grassland specialists, which you might have guessed based on the backgrounds of these pictures. They depend on native grasslands for survival, but more than 90% of these habitats have been converted for other uses since colonisation. And because of this, they're now thought to be fewer than 1,000 individuals left in the wild. So there are lots of organisations across the country working on the recovery of the species, and for my part, I was working on the conservation genetics, which involved working with collaborators from different organisations like Parks Victoria, Zoos Victoria, Taronga Zoo, Bush Heritage, government departments in 4 different states to collect samples for genetic analysis. This work meant that we were able to answer really important questions like, do plains wanderers actually wander? Are they moving around or are they staying put? Are populations genetically similar or dissimilar? And what are the levels of genetic diversity like? None of these questions have been asked or answered before, And having answers to these questions meant that we could make decisions about which birds should be paired in captivity, where releases could happen, and how populations in the wild should be managed. It was a very rewarding experience to be involved in work that directly impacted the conservation of a threatened species. And it was also a really interesting experience for me working with a really diverse group of stakeholders in this project. As my fellowship was wrapping up, I got the opportunity to join the EPA working in the data analytics team. This gave me the chance to see how I could apply the skills that I'd developed during my PhD and postdoc in an industry setting. The broad remit of the team was to carry out data analysis that supported the operations of the EPA and to inform decision-making. This meant that in reality, I got the opportunity to work on a really broad range of projects, like mapping levels of contaminants in soil across the state, or creating platforms for businesses to register changes as environmental legislation got updated, or analysing how patterns of disposal changed for different substances following the 2020 bushfires and during the progression of the COVID pandemic. Working within this team was a really incredible learning experience for me. In academia, any data analysis that I did was directly related to the specific questions that I was asking. I would have collected the data myself, so I was familiar with what I was looking at, and I had expert knowledge of the system that I was working in. In this job, however, it was completely different. I was often working with data that had been collected by someone else, not necessarily in a systematic way, and often related to a system that I had no experience in, like soil biology or waste transport. This job, I found, helped me to build a really strong foundation in the techniques of working with messy data. At the same time, it was also really interesting to see the sorts of transferable skills that I brought into industry from having worked in academia. For instance, having experience in landscape ecology and genetic spatial analysis put me in a really good position to work as a geospatial analyst. After a while, I did start to miss working with biodiversity data. So when I had the opportunity to join the Atlas of Living Australia as a data analyst, I jumped at the chance. I primarily work on the EcoAssets project now, which brings together environmental data collected from 3 national research infrastructures, the ALE, IMOS, and TERN. Large amounts of data are collected every year on different aspects of the environment, but these data are not always comparable, accessible, or provided in formats that are suitable to use in reporting. A lot of the work that I do revolves around standardizing the formats of these data resources and aggregating them into data assets that support Australia's environmental reporting needs, such as the State of the Environment report. One example of this is starting with over 100 million occurrence records in the Atlas and filtering and aggregating it such that we can answer questions that will help us understand what's happening in the environment, whether that's at a local scale or nationally. Questions like how many threatened species occur in terrestrial and marine bioregions, or how has the number of invasive species changed across the years in different parts of the country? Do protected areas have more species diversity than non-protected areas? These are examples of some of the questions that we've addressed with the datasets we've created, and we're continuing to work with research partners to understand different types of reporting needs. In addition to playing around with absolutely enormous datasets, I also get the opportunity to create visuals that illustrate some large-scale patterns, like this one. This shows the proportion of species occurrence records in each bioregion and each time period that derive from human observations, which might be a field survey or citizen science, for instance. And this is in contrast to specimen-based data. So in this image here, marine and terrestrial regions are on different color scales, and the darker colors show higher proportions of human observations. As you can see, that those darker colors become a lot more prevalent as time passes. These data are all available on the EcoAssets website, but making figures like this is a really nice way to communicate changes over time and space in a visual manner, which can sometimes be more intuitive to understand and facilitate the process of science communication to different audiences. This has very much been an overview of some of the projects that I've worked on over the course of my career. And although they're all quite different, I've really enjoyed working on all of them. And reflecting back on them, I found that it's the process of solving interesting problems, whether that's trying to understand what a species is doing or figuring out how to get the most out of a data set, that has kept me really engaged in my work. Thank you very much.
Thank you, Sandy. That was fantastic, and what a beautiful set of slides as well. I love, I love your palettes that you've chosen there. As before, we're going to hold most of our questions to the end. I will say though that Sandy and I both work at the ALA, and we work together quite closely. And there's a lot of things in there that I had no idea about, despite having interviewed you for your current job. So it's quite interesting, isn't it? How you think you know someone, and then you're like, oh, I did that. That's really cool. We should talk about that sometime. And will. So.
Martin, we lost you for a sec.
Sorry, probably the internet in here. I was just going to hand over to Casey if you wouldn't mind sharing your slide.
Thanks, Martin, will do.
Okay, that's working perfectly. Over to you, Casey.
Beauty. All right. Hi, everybody. My name is Casey Kershoff. I'd just like to acknowledge that today I am on Gundungurra country and I pay respect to elders past and present. So today I've got a little bit of I guess my story is quite fledgling. I'm still a PhD student, so I'm really on in my early on in my career. So where am I now? I'm a part-time PhD candidate in ecology at UNSW. So I'm researching climate change impacts on Australia's alpine flora, while also parenting a toddler and having a baby on the way. Also another project is a citizen science project called the Environment Recovery Project, which I took a major detour into after losing our home in the 2020 bushfires. So this project has gathered 28,000 observations since we started in 2020, together with the big bushfire bioblitz that we ran in conjunction with the project. And then a highlight so far is that the Environment Recovery Project team and I have been shortlisted as finalists in this year's Eureka Prize for Innovations in Citizen Science. So how did I get here? So I will very much be happy to say that I was not a very studious public high school student. I was a little bit more interested in maybe not going to class and going surfing down the coast, growing up on the south coast of New South Wales. So I really never had any aspirations of going to uni. So I bounced around after high school in the ski fields, working in real estate, working in hospitality, just kind of getting a whole mix of life experiences. After meeting a guy who has now turned into my husband, I was encouraged to start university because he picked up that I had a bit of an interest in plants, which was bang on, I do, and I'm now kind of more trained as a plant ecologist. So I head along to uni at the age of 25 to start a Bachelor of Arts. So I totally agree with what Mel said earlier. It's really cool having a grounding in the arts, coming into that as bringing that training in the arts and the humanities into science is such a wonderful thing. So during that first year of university, I enrolled in 2 elective science courses. One of those was ecology and another was earth science. And immediately I was like, okay, I need to change my degree to a double degree because this science thing is something I'm actually really interested in, which kind of blew my mind because I never even really did any science in high school. So here I was now at university and taking science courses. Never something that I thought that I would actually do. So it was during that time in my undergraduate, after shifting to a double degree, I started using the Atlas of Living Australia. So at first that was during coursework. So we were using it in our classes. We were learning how to use the biodiversity data that was available for mapping, threatened species occurrences, that sort of thing. And then not long after, I started dabbling in citizen science as a citizen scientist. So I would manually upload my observations of mostly plants and fungi into the Atlas. and I actually accumulated quite a number of observations, and that really has shaped the way that I've ended up doing things, into, where I'm at today. And I have extensively used the ALA in my doctoral research, and also in teaching and demonstrating undergraduates in ecology and conservation as I've moved into that during my PhD work. So my PhD work ties in a little bit with, some of that work that I did after leaving school that I thought would never really have an impact on my life. So working in the ski fields, I've turned into an alpine ecologist. I guess once you're a bit of a ski junkie, you kind of stay that way. So a huge part of my research has been doing a winter field experiment. So my love of skiing and my love of alpine plants have met really nicely. You can kind of see on the left here a nice hole that I have dug in the snow, one of many, many, many holes dug over the course of a number of winter seasons. So I was doing a microclimate experiment to see what happens when the snow cover is minimized or completely removed during the winter. So on those plots, we had little microclimate loggers called eye buttons measuring the temperatures. So we had plots that had snow completely removed, we had plots that had a little bit removed, and then plots that were left as is, just to see the differences in temperatures. So when you take the snow off, it obviously gets a whole lot colder, and then the plants can have stress responses relating to that exposure to freezing during the winter and early spring. So that was a really cool thing to do, going out and being in the field. It was a year-round field experiment, so a lot of winter work, but then also going out during spring for melt and during the summer walking around the mountains. I also use a lot of herbarium and historical data and climate records. So the Australian Virtual Herbarium, which is tied in with the Atlas of Living Australia, a lot of that data is used in my herbarium study. So it's another way of looking at alpine phenology. So phenology is the timing of events in a biological organism's life. So in this case, I'm looking at wildflowers, which is not the worst thing to be looking at. If you're up in the beautiful mountains, you can kind of see some of those wildflowers there on the right-hand side. So looking at things like a plant's flowering or fruiting earlier as the temperature warms. And during my PhD candidature, I've also I had taken a bit of a role in science communication and citizen science as well. So this is a little bit old hat now, but back when Instagram was kind of in vogue and the old style posts, that was a really nice way of conveying the beauty of the Australian Alps with little snippets of science. It was mostly alpine flora of Australia. So there was, there's a little invertebrate down here, but it was mostly beautiful plants up in the mountains. Oh, sorry, we've got an interruption. Sorry. Okay, sorry, if you hear my toddler, I apologize. Using these social media platforms as a way of conveying science and getting people interested in different types of plants that maybe they're not interested in, becoming aware of issues surrounding the Australian Alps, for example, the way horses and other feral animals were managed in that area. So the second part of how did I get here kind of forks off quite dramatically from the Australian Alps and the beautiful wildflowers. So this is a picture of my backyard and my house on the 4th of January 2020. So we were directly impacted by the 2020 bushfires. My husband and I have a 5-acre property up in the Southern Highlands. So our entire property and home burnt down. And it was at that point, having my experience as a citizen scientist and my love of kind of uploading observations, which had turned into iNaturalist at that point, I wanted to see what was coming back in my own property. So 4 acres of our property is just beautiful, dry sclerophyll woodland. So I wanted to see what plants, what animals were going to be coming back, were there any traces of life still out there? So from that, talking to colleagues up at the University of New South Wales, we decided, well, a lot of people were impacted by those bushfires, not just me, of course. So maybe this is going to be a bigger project. Maybe we need to make this something that everyone who had been impacted in any way by the bushfires could add their observations into. And from that, we started the Environment Recovery Project on the iNaturalist platform. So that's basically it. It was a project designed to be as easy for people to use as possible. We had a few observation fields that people could fill out if they wanted to, relating to maybe landscape variables such as how high up the trees had the fire burnt. So how black is the trunk of the tree? What does the canopy look like? Was it completely consumed, or was it just scorched or still green? So we gave people a couple of really simple things that they could fill out, or if they wanted to, they could just dump a photo which is geotagged and then has the location, the time, and then the species observation, which would come in later. So that's gone pretty well over since we launched in the 30th of January 2020. We've had over 20,000 observations come in, which has been really cool. So that 3,121 is the number of species that have been observed so far. Okay, sorry, I'm jumping the gun here. So over 3,100 species. So we've over 1,000 project members, and of those, we've had about 448 people that have contributed. And right across Australia as well. Originally, we were focused on the East Coast. However, there was obviously a lot of burn happening on Kangaroo Island, and then subsequent bushfires in the Perth Hills. And then we've even had some, I think that's a macrozamia from the McDonnell Ranges out there in the central from a bushfire there. So we've actually opened the project now to all bushfires that have happened either before the 2020 mega fires and any future fires as well. So the project is still open and collecting data. There's still people actively contributing to it because bushfire recovery is a very lengthy process. So from the bushfire recovery, the Environment Recovery Project, this has led into a second project which was just fantastic to work on, which was the Big Bushfire BioBlitz. So myself and colleagues from the designed 3 events in conjunction with the Atlas of Living Australia to be held across New South Wales. So the first of those was up in the Blue Mountains, an area that was extensively burnt during the 1920s and other fires. So we had a BioBlitz up at the Govetts Leap area, and there was a lot of surveys going out from that point with a whole bunch of different survey leaders, people that were experts in fungi or birds or plants. So it was a really nice way of getting the community together to come and maybe see what was coming back in their own backyard. And this was a smoking ceremony by Lexodius Dad, who's a wonderful Darul Elder. Our second event, which was not rained out like our first event on up at the Blue Mountains, was down at Marmarang National Park, also an area that had been significantly impacted by the bushfires. We had spectacular weather. Again, the same sort of setup where we had a whole range of surveys going over the course of the weekend. There were a lot of really cool rare and threatened species spotted at this particular event, greater gliders, glossy black cockatoos, lots of strange little mollusks in the forest, a couple of rare plants. So again, a really great way of engaging the community, and we had a really particularly good turnout at this event. And so the third event, which I'm really quite bummed that I personally didn't get to attend because our bushfire rebuild was finally complete and we got the keys on this weekend. So I didn't get to go and meet Costa, who dropped into the third event, which was at Crowdy Bay and Duragan National Park up on the mid-north coast. But that was really by the team here, Thomas Masagio, and a few others from the Centre for Ecosystem Science. And we've got Erin here, who is one of the ALA people who was working on the project. So all up the Big Bushfire BioBlitz, over the course of those 3 events, gathered over 8,000 observations. About 1,800 species were seen. There were about, well, I'm not too sure of the number of participants, a couple of hundred people that had participated, and then also quite a few more people that had been coming into iNaturalist to make sure those identifications were firmed up. And so together, the work of the Environment Recovery Project and then the subsequent Big Bushfire BioBlitz kind of put us into this position, which is a definite highlight for my bit of a strange career so far, is that we've been nominated as finalists for this year's Eureka Prize in Innovations in Citizen Science. So that's really cool, because that kind of talks the value of citizen science data, that people can go out and be observing things in their own backyards, and it actually does have direct, it's directly useful for biodiversity research, especially if we're looking in the context of bushfire recovery. So on the team here, we've got Richard, Thomas, myself, Will, and Mark. So yeah, so that's pretty dang exciting, I must say. So just to wrap up, I guess because I'm so early on, I'm still doing my PhD. It will be finished one day. I've been doing a lot of side projects, as you can tell, in citizen science, and there's a bunch of other stuff as well. But like many things in ecology, my career paths in STEM are often non-linear, and that's okay. There's no one way. There's no one direct route usually in doing things, and I think that's it for me.
Thanks, Katie. That's a fantastic talk. And look, now we've got 20 minutes or so to speak. And if you don't mind, Casey, I might pick up on something in your talk first, which is, there's so much that I could raise here because it was fascinating. So the things you discussed, but one is, it occurred to me that we've all talked here about crossover between the things we're personally interested in and the things we choose to study, and that sort of, Sandy mentioned intellectual curiosity as being a driver for the choices she made. In your case, to do this bushfire research is a pretty extreme crossover from your private life and into your professional life. And was that cathartic or helpful for you in dealing with those challenges?
Oh yeah, no, absolutely. So that was my way of, I guess, I saw those little green shoots in my backyard a couple of days after the bushfire, and I was like, right, that's what I'm going to hang my hat on and focus on. In amongst all of the, the horribleness of the bushfires, I'm just going to literally watch the grass grow. And that's a sentiment that's now been shared with a lot of the participants that were, directly impacted, that had maybe their own properties burned down. Participating in the project has been a way that they can focus on positive things, and also, learn new things about their property that they maybe didn't know, or maybe not even their property, but their local national park or area that they're passionate about. So yeah, it's been a nice little thing to obsess on.
It's funny, isn't it, to find science is so much of for those of us that engage in day to day, it is part of our passions, and it's nice to share that, I suppose, more broadly through a citizen science initiative like that. So look, we've got a few questions coming on the channel. I'm going to try my best to summarize them. I hope people listening at home feel that I do a good job of that. There's always more than I can ask to the whole panel. I guess. And anyone feel free to jump in on this one. It seems in each case that there's a combination of trying to train in skills that you thought were interesting and valuable. So in some cases being a bit strategic about perhaps the things that you went to learn versus just following interests. And to what extent did you choose to do things because they were potentially useful or employable versus just things that you took an interest in? Perhaps Mel, would you like to comment on that?
I'd just say with collection management, it's always been really interesting, because I actually did, part of being an assistant collection manager in the early part of my career. After volunteering, I did that. But then when I went away for 7 years, I got so many different skills that I brought back that I didn't necessarily at all think would help. But there's just, different kinds of strategies, different kinds of management skills, even just writing skills and, social skills. Like, there's so many things. I think the thing is that you can never discount what you do, even all the little part-time jobs. I mean, from tomato weeding to, you know, to toilet cleaning to everything else, like everything you do. I think the thing is to not ever think of it as a dead-end job, because anything you do might one day contribute to something else that is really what you want to do. So I just think it's always been very positive. I actually didn't, a lot of the things I did were not directly, other than volunteering, we're not specifically trying to build towards a career, but they did all help me in some way. So that's what I think. And I think it sounded a bit similar for the other two, but I'm not.
What about you, Sandy? You've trained in some extremely technical fields. You know, I know almost nothing about genetics and basically couldn't follow those conversations if you were to have them with me, and yet we do similar jobs. Is it simply a case of intellectual curiosity and seeing where you end up? Or is
it was just curiosity about the things that I was looking at the moment without necessarily having an endpoint in mind, which maybe is a reflection of me. I think a lot of it was a leap of faith that there would be something, because one thing I realized looking back is that I didn't know what a career pathway would look like. I kind of knew I liked science, even though, and you know, this rings true with what Casey said as well, I was terrible at it in school. I was interested in it, but I was never very good at it. And I kind of liked it, and I thought, you know, I don't know what a career pathway looks like. I might as well do something I'm enjoying, and we'll see where that leads. And that's kind of what it's been like at every point. I mean, I started an honours year because I was really interested and I didn't know what else I should be doing. Seemed like a good idea at the time, which maybe is something 20-odd-year-olds do. And it led to the PhD, which I wasn't expecting. And the same for the postdoc. I wasn't necessarily expecting to come out of a PhD and go into postdoctoral research. It just, right place, right time, right experience, I guess.
Yeah, that very much my experience as well. And I don't know, Casey, you mentioned at the end that one day you'll be finished your thesis. I think it took me 6 years from enrolling to ending in my doctorate, which I probably shouldn't tell my boss. So if that's any consolation, but it seems like you very much followed your interests and just saw where they led. Is that a fair description?
Yeah, absolutely. I totally agree as well with everything that's been said. So a lot of the jobs that I've done in the past, you don't think too much of it at the time, but now I can reflect and be like, oh, okay, no. So like when I was a cleaner, I can use those skills. And when I worked in real estate doing marketing, I actually now can make a poster and win an award at ESA because, like all of these little things that you do, they're maybe not as trivial as you think they are, and they can be really beneficial. And it can be cumulative, those skills that you gain. But yeah, I can also be quite impulsive and just kind of do what I feel like doing, which has kind of led to some really cool things. So that, the Citizen Science Project, and just doing something that I'm interested in.
It's hard to argue with being nominated for a Eureka Prize for saying that was, a misuse of time, even if it wasn't exactly what you planned to do at the time, perhaps. So I did laugh at this question, so apologies to the person who asked it, but someone did specifically ask how they could get on a research vessel. Now, how do I get a job on a boat? I guess it's an interesting point, though, isn't it, that you said that that your pathway into museums was a little winding, and is the museums a growing sort of sector, and is it routine for people to get voyages as part of that?
I think it depends on the institution or the museum at the time. So at the moment, we've had Chief Scientist Tim O'Hara has been pushing for a few voyages in the last few years. So we've got another voyage coming up. So we've had a few voyages in a row. And I think the same thing happened back in the 90s. There were a whole lot of surveys and that, so there are quite a lot of voyages in a row. But it kind of depends on who's there at the time. So it's not always guaranteed, but there are places like CSIRO and that do have ongoing voyages, quite often. And there are certainly institutions all around the world that have a lot of voyages. For me, to be honest, my very first voyage was from volunteering, because I volunteered with that sea cucumber mentor, studying sea cucumbers with him and helping to identify things. There was actually 3 of us. There was myself and him and a younger high school girl actually studying these sea cucumbers in our spare time. And when when an opportunity came up with the British Antarctic Survey to go to Antarctica, the mentor decided he was too old, the other girl was too young, and I was just the right age. So it was really just luck. And then once I'd been on it once and people knew I was capable and would really work hard, then I was invited back and back and back. So I think really making the most of any opportunity that comes to you, and just always and like I said, when I was in the Falkland Islands, like I was there for the Shackleton Scholarship there, but then while I was doing that, I was volunteering on the weekends doing other stuff. I mean, why would you waste these opportunities? You're in such an amazing place. You might as well, do all the work you can and meet as many people as you can and try and have as many chances to do things as you can. And I guess that's what I would suggest. I mean, there are definitely different institutions that may do more voyages than others, but we've been very lucky in the last few years. So, yeah.
Certainly some very impressive ships. We're actually part of the same CSIRO unit that runs the Investigator, I believe it is, the ship that runs out of Hobart.
That's the ship, yeah. That's the one we're going to be back on again very soon. So, yeah.
Oh, really? Oh, wonderful. I've avoided it this far myself. I think I'd get very seasick, but I'm glad that other people can make the best of that and enjoy that and do some cool science at the same time. Look, we've all talked here about ironically, perhaps underachievement at earlier stages in our career or at school, and the sort of winding pathway that brought us here. We've had a question from Ron in the chat saying, would we have any advice for someone who wanted to change careers, perhaps later in careers or perhaps in their 40s or 50s, into biological science? Any perspectives on that?
Do it. I mean, I
no regrets then, Sandia?
This is a slightly facetious answer, but I mean, I assume if the person's asked, it means they've thought a little bit about it, and if they're thinking about it as a serious choice, then why not take the leap? I mean, people change careers all the time. I don't feel, I mean, in my talk it was a linear timeline, but I don't feel like it was particularly linear, and it doesn't all feel like it was in the same field. I've worked on very different things, so I don't think those leaps are necessarily bad or even unusual as such.
Yeah, I do remember reading a book on research to advice to scientists by Ed Wilson, actually saying that people think that that they just don't have skills in a new field, but actually they're bringing their entire background with them. And it's often quite surprising, the insights you can gain because of your background rather than in spite of it. And I tend to agree with that. I guess as well, an interesting point to think about here is that we're all in, I remember being in university and working there, and this sort of perceived split between working in a university as an academic and other fields. And yet here we are, 4 scientists in different institutions with different sort of funding arrangements and different structures, and yet all still doing very science-like activities. I don't know if anyone else has made jumps between those institutions quite as much, but it's amazing where you can make a contribution. So now I'll just see if we've got any. Now here's a final question I've got as well from the chat. Do you have tips for people who are less able to find a career path, who can't work full time, for example, or are not, or may perhaps have disabilities or health issues? Has anyone have any insights on differently abled people's experience in the workplace in science?
I'd just say, I mean, I don't have actually. Casey should probably speak to being a mum with having toddlers running through.
Yeah, how's that been, Casey?
Yeah, I mean, I work 2 days a week, so I'm tapping away. I am an able-bodied person, but I know that depending on people's different abilities, there's still a lot of things that can be done. Bioinformatics is a great thing. So that's where, the data of the ALA, if somebody has mobility issues and can't actually get out too much, you can be a scientist and be diving into the data like we've seen today. And then, you know, there's still a lot of field research that can be done as well, for people with different abilities. So yeah, I think, you know, don't let your ability hold you back. And there's always going to be a supportive mentor out there that can kind of take you under their wing to help you find your feet or find your way. So yeah.
Yeah, thanks, Katie. Look, as someone who works part-time and also has a toddler running around at home who would have been, let me tell you, considerably louder than yours was if I was sitting in the same room with him. Yeah, I'd second that, actually. Assuming you can find people who are willing to be good mentors and set up a good workplace, and I think it's a lot of people these days. I don't think it holds you back in the least.
Yeah, I would actually just say, just don't assume that you can't do the job, because I just think that there are, I mean, there's things like Digivol and that, again, what you were saying, Casey, like things that people can do. A lot of those things are voluntary, of course. So I know it's harder to get employment, but if you're doing, but if you try those kinds of jobs, then they might give you the skills that you need for other jobs. And, you know, there are definitely things out there that people can do. So don't be afraid to ask, I would say. Yeah.
Oh, definitely. And lovely. Well, thank you again for your time, everyone. I think we'll leave it there. We've gone through most of the questions that I could come through from the question channel. For those at home, thank you for tuning in. We run these roughly quarterly. Keep an eye out for the next one, which I imagine will be in October or November. Thank you for your time and have a lovely day.